Hello everyone, this is Senior Biotech Analyst John Vandermosten. Welcome to our channel that educates the life sciences investor on exciting advancements in drugs, biologics, and devices. For more content and news like this, subscribe to our channel below and like or share with others. Tiziana's lead candidate, Foralumab, targets the CD3 receptors on T cells, helping temper the activity of the immune system and to reduce inflammation. The mechanism opens up Foralumab to a wide variety of diseases, including COVID, intracerebral hemorrhage, and other inflammatory disorders. Welcome back to our conversation with Dr. Matthew Davis, Tiziana Life Sciences Chief Medical Officer. Dr. Davis, what has Foralumab been able to achieve in long COVID patients?
So far, we see enough data out there in the literature to believe that microglial activation is an important part of long COVID. Now, in patients with COVID, patients with multiple sclerosis, we've seen that Foralumab would take activated microglia and return them back to a baseline non-active state. So it's our intention to take patients with long COVID and ask a simple question. 3 months of exposure to intranasal Foralumab, will that in itself return the microglia back to a non-activated state?
And I think probably a lot of people wonder what the difference is between long COVID and regular COVID. I think we understand what regular COVID is, but how is long COVID different?
There's a lot of definitions, and I think the simplest way of looking at it is a long-term effect after COVID, after all the symptoms of COVID that we know are gone, that are adversely affecting a patient. And the number one symptom people call is brain fog. And this is not letting people go back to work and function in a way that they want to function. This is costing the healthcare system over $1 trillion in estimated costs. And it's a huge impact in people's lives, their ability to function. And it's a very interesting target because of the research already done looking at the correlation between microglial activation and long COVID itself.
And just for a statistic for everyone, we looked at a recent Kaiser Family Foundation study that found about 5% of the population actually has long COVID. So that's pretty substantial. I don't know if you want to elaborate at all on the prevalence of it.
I don't think anyone in your audience or you and I don't know someone who doesn't have it. Mm-hmm. So it's unfortunately ubiquitous right now in society, and those numbers seem right to me.
Okay. You guys conducted a COVID study in Brazil a little over a year ago, I think, maybe a year and a half ago. The results from the study came out more recently, and they were rather technical. There were a lot of series of letters there and receptors and things like that that were discussed. Can you summarize it though in a less technical fashion for the audience?
Right. First of all, kudos to the principal investigator who in 2020 went to Brazil with basically a biohazard suit in the beginning of this.
Right, because we didn't really know what the risk was.
And just an incredible warrior researcher. I just want to give credit to her. And this was just recently published in the prestigious journal of PNAS. Summarizing the article, intranasal Foralumab is going to change T cells, which are a regulatory aspect of our immunity system. Sometimes they up activity throughout the body, sometimes they will mitigate the activity throughout the body. And it'll take T cells that are in your cervical lymph node, squirt, and it will turn them into a Treg, the regulatory aspect of the T cells. Those T cells will go across the brain in this specific example, and they'll go to microglia, which are activated, which means they're turned on. They are in the mode in which they're going to act as an inflammatory agent. And it will change the microglia back to a non-activated state. And this study looked at this information in a lot of different and technical ways. By the end of the day, the genes are changing. Some genes that are pro-inflammatory are turning off. Some genes that are anti-inflammatory are turning on. And the clinical outcome on the patients with COVID, they were able to clear their lung infections vis-a-vis CAT scans. They recovered quickly, and there was a difference between a placebo, and there's even a difference with giving dexamethasone. So it was a really well-done trial that show that in COVID with these patients and these patients, they got benefit. But I think more importantly, it correlated with what we saw with healthy normal volunteers and in our multiple sclerosis patients, that the genes that we wanted to be turned on that were anti-inflammatory were turned on. The genes that were pro-inflammatory were turned off. Very exciting research.
So if you, in further studies here, what are some of the biomarkers that you'll be looking at to measure success?
I would state that pretty much everything that you've read about, New England Journal of Medicine, Nature, et cetera, that you can imagine that is blood biomarkers or CNS biomarkers are going to be in our phase 2A. So there's no stone being left unturned. We're also going to be looking at a large amount of the interleukins that have been associated with everything from inflammasomes and inflammatory reactions to other interleukins that you would prefer not to see that are pro-inflammatory. And we're going to get an excellent picture of what's the difference between our product at 2 doses versus a placebo in a 3-month time period. Very exciting data.
Yeah. Well, thank you, Dr. Davis. Again, we've had Tiziana Life Sciences Chief Medical Officer, Dr. Davis, with us today to speak about the long COVID opportunity for the company. Tiziana Life Sciences, ticker symbol TLSA.